So students, let's start with your queries first. So if you have any questions, you can ask. So in the previous class, we talked about IgA. Okay. So now we have talked about three classes of immunoglobulins, right? IgA. Before that, we talked about IgM, and before that, we talked about IgG. Okay. So in this class, we will talk about IgE and IgD. Okay. First, I will talk about IgD because there is not much to tell. Okay. So I'll just first tell about IgD and then we will talk about IgE, and again I will not talk about IgE in much details, the mechanism and all, because that we are going to learn when we will talk about the hypersensitivity reactions. Okay. So let me share the slides. Okay, so immunoglobulin D has very low serum concentrations. Okay, so if you compare it with IgG, IgM, or any other type, so IgD has very low concentration. Okay, 0.2% of total immunoglobulins. Okay, so just imagine or just compare 80% IgG and IgD 0.2%. Right? So you can imagine how less amount of IgD is present in the serum. Okay, and this immunoglobulin D is expressed on B cell surface. Okay, so when we say it is expressed on B cell surface, it means like this is your B cell, okay, or this is your cell, and it is expressed, so it is there on the surface. Okay, so it is expressed outside. Okay, and only two types, IgD and IgM, they are expressed on B cell surface. Okay, so IgD is one of them which is expressed on the surface of B cell. I hope this is clear to you. Uh, so suppose this is your B cell, okay, and on the surface of this cell, IgD is expressed, okay, and it is always expressed like this, where the Fc region is bound to the cell and Fab region is open, where the antigens can bind. Okay. Another thing, it's a puzzle in immunology since its discovery in 1964, and that's why we don't know much about it. Okay, it's still a puzzle. We don't know exactly what it is doing in our body. Does it have any role in binding the antigens, or does it have any regulatory functions? Okay, so one of the function that is known is the function in activation of B cells. Okay, so IgD helps in activation of B cells. Again, we do not know the proper mechanism. It was also found to bind basophils and mast cells, okay, and activate these cells to produce antimicrobial factors to participate in respiratory immune defense in humans. Okay, so this is another role which have been found. Okay, it basically activates the basophils and the mast cells. Okay, and so we have not read yet, but when these cells, basophils, mast cells, they get activated, they release their granular content. Okay, so when there is some parasitic infection or some any kind of infection, in that case, these cells, basophils, mast cells, they release their granular content, okay, and that helps in the killing of the pathogen, okay. So this is another role which has been uh, like assigned to IgD, okay, and still a lot of research is going on. Other biological activities are not known, so we actually don't know much about IgD. So the good thing for you is that you don't have to study much of IgD, so you can now surely focus on the other four types. Okay. Okay, so let me again show you. Okay, so as I said, IgM and IgD, they are membrane bound. So I'll just show you a video which talks about both IgM and IgD. Okay, so that will make things more clear to you. Okay, so this is the pentamer of IgM, and IgD is present in its monomeric form. Okay.
GD. Both of these are membrane-bound immunoglobulins. They are found on the surface of mature naive B cells. Let us now study their structure, properties, and function. Immunoglobulin M constitutes about 5 to 10% of the antibody concentration in the serum. It occurs both in monomer and pentamer form in the body. As a monomer, IgM is present on the surface of B cells. This IgM is known as membrane-bound immunoglobulin M. IgM as monomer consists of 2 mu heavy chains and 2 light chains. Each heavy chain contains 1 variable domain and 4 constant domains. Now let's look at IgM as a pentamer. As a pentamer IgM, it is found in the secretions. In pentameric form, 5 IgM monomers are arranged in a star-like shape. In this shape, the antigen binding fragments are outwards, and the Fc fragments are towards the center. These monomeric units are held together by J chain. Recall that J stands for joining, and it is a glycoprotein. This J chain is responsible for the polymerization of IgM into pentamer. As you see in this image, the theoretically IgM pentamer has 10 antigen binding sites, but only 5 or 6 of them are available for effector functions because of steric hindrances. Still, this number of antigen binding sites is maximum among all antibody classes. Immunoglobulin M is mainly found in the blood. It is unable to move freely because of its large size, and therefore, it is not found in other body fluids. Half-life of IgM is about 5 days. It is the first antibody that is formed by the fetus. Also, it is the first antibody that appears during an immune response. IgM is the most efficient antibody which activates classical complement pathway. This is because of its large size with multiple antigen binding sites. A single IgM antibody can initiate the activation. IgM is very important defense against bacterial diseases. These antibodies are very effective neutralizers and prevent pathogens from binding to host receptors on epithelial cells. They do not act as opsonins, neither they play a role in antibody-dependent cell-mediated cytotoxicity. This is because specific Fc receptors to IgM occur rarely on the surfaces of leukocytes. Let's now talk about immunoglobulin D. Structurally, IgD is a monomer. It has two delta heavy chains and two light chains. Each heavy chain has three constant domains. The hinge region of IgD is longest among antibody classes. It mostly remains membrane-bound. IgD is one of the main receptors on mature B cells. It is found on the surface of B cells in addition of IgM, which together form the part of B cell receptor complex.
So students, you can see here that IgD and IgM are membrane bound, right? And when they are membrane bound, we put a small m in front of it. Okay, so this is membrane bound immunoglobulin D, membrane bound immunoglobulin M. Okay, and you just saw in case of IgA, we put small s and capital S, right? To denote whether it is the serum one or the secreted one, right?
IgD make up less than 1% of the antibody in the serum. It has a half-life of only 3 days. Its functions are unknown, but it seems that they play an important role in recognition of antigens by B cells and in signaling B cell to start antibody production.
So students, is everything about IgD clear? So not much. Any questions? Okay, so then we can start with IgE. Okay, so coming to the fifth class, IgE. Okay, so the fifth class is IgE. So we have now talked about IgG, IgM, IgA, IgD, and this is the fifth one, which is IgE. And this antibody was identified by two scientists, K and T Ishizaka. Okay, so Ishizaka's they identified IgE in 1966. Okay, again, IgE also have very low serum concentration. Okay, so the main role of IgE is, so see what happens. We always say IgE is associated with allergies. IgE is associated with hypersensitivity, but in reality, that's not the only role it have, and that's not a good role, right? So IgE in actual have a role in fighting against the helminths and nematodes, right? So if there is a helminthic infection, parasitic infection, there will be a high amount of IgE. Okay, but IgE also participate in immediate hypersensitivity reactions. Okay, now what is this immediate hypersensitivity reaction? We will discuss later. But in patients of asthma, anaphylaxis, hives, you will see a very important role of IgE, and we will just discuss briefly how this happens. Okay, what IgE does to cause hypersensitivity reactions. Okay, and this IgE, it binds mast cells and blood basophils through which receptor? The Fc epsilon R receptor, right? I'm sure you now understand this. Fc is the Fc region, epsilon is the IgE heavy chain, okay, and R is the receptor, okay. So IgE binds to the mast cells and blood basophils through Fc epsilon R, okay. So where is this receptor present? It is present on the surface of mast cells and basophils, okay. And when IgE binds to these receptors, it causes degranulation, okay. So degranulation means the whatever the granules contain in basophils and mast cells, uh, those mediators are released from the cells, okay. And I'll just show you uh, this process, but before coming to that process, I'll show you a small video on IgE. Okay,
Structure, properties and functions of immunoglobulin E. Structurally, IgE is a monomer. It contains two epsilon heavy chains. Each heavy chain has four constant domains like IgM. IgE is found in trace amount in serum. It is the least abundant immunoglobulin in serum. It has a half-life of 2 days. IgE does not participate in complement fixation, opsonization, etc. But it is very crucial antibody because it play important role in allergic reactions. Also, it is important in defense against parasitic worms. Let's discuss the role of immunoglobulin E in our body. First, let us discuss role of immunoglobulin E in defense against parasitic worms. When there is an invasion by parasitic worms, the T helper cells secrete cytokines such as interleukin-4 and stimulate B cells to secrete IgE antibodies. The concentration of IgE antibodies in the serum rises. These antibodies then coat the surface of the worm by binding to their surface antigens. These bound IgE antibodies are recognized by the eosinophils. This recognition is mediated by specific Fc receptors present on the eosinophils. These receptors recognize and bind Fc region of IgE antibodies. Once bound, they undergo degranulation. The released granule content destroys the parasitic worm. Let us now look at the role of IgE in allergic reactions. The immune system of some people is sensitive to substances such as pollen, nuts, dust mites, etc. These substances are known as allergens and ordinarily harmless in most people. But the immune system of sensitive or allergic people recognize these substances as dangerous. When there is an initial or first exposure to an allergen, B cells are activated. These B cells differentiate into IgE-producing plasma cells. These IgE antibodies attach themselves to the mast cells and basophils by their Fc regions. Now, the antigen binding sites of these IgE antibodies are free. When there is second exposure of the allergen binds to these antigen binding sites of IgE antibodies. This binding of allergen to IgE antibodies results in the cross-linking of IgE antibodies. This cross-linking sends signal to the cell and results in degranulation. As a result of degranulation, there is a rapid release of preformed mediators from the granules. For example, histamine, heparin, etc. These mediates further result in vasodilation, increased vascular permeability, smooth muscle contractions, etc. This reaction is known as allergic reaction. Thus, elevated levels of IgE antibodies in the body of diagnostic significance. Elevated levels of immunoglobulin E indicates the possibility of allergic reaction or parasitic infection.
So students, as I said, uh, the role of IgE is one against the parasitic infections. Okay, you just saw that those IgE molecules, right, from the FAB region, they will go and bind to the parasite. Okay, and the FC region, it will go and bind to the eosinophils, right? Now, when this binding occurs, the eosinophil releases its content, okay, and the content that it releases, it goes and binds to the parasite, okay, and the parasite is killed, okay. So this is the important role of IgE against parasitic infections, okay. But in some individuals, right, in some susceptible individuals who have some genetic uh, problem, right, so in only those people you see allergic reactions, okay. Now one person may be allergic to a particular thing, another person may be allergic to another thing, okay. And I'm sure we all have experienced these allergic reactions, right? Sometimes what happens, suppose you have touched a particular substance, just for example, a particular leaf, okay, and there may be redness on your skin, okay. Some people are allergic to the rubber, right? Some people are allergic to some food stuff, right? Some people are allergic to eggs. Now, it's a very big problem for them. The people who are allergic to eggs, they cannot take the vaccines, okay, because when we develop vaccines, the viruses, they are grown in eggs, okay. So that is again a problem. So I'm sure you know, like some of you are allergic to dust particles, some of you are allergic to some components in the shampoo, right, or skincare product. So uh, people are allergic to a lot of things, okay. And again, I'm not going to discuss the process here, but um, ultimately it's the IgE which is mediating these allergic reactions, okay. And how it mediates, I'll show you with the help of this diagram. Yeah, so if this picture is clear, so this is your mast cell, okay. So we are taking an example of mast cell, okay. Now what happens, this is an allergen, okay. So allergen is an antigen, okay, but we appropriately call it as allergen because in some people it is causing allergy, okay. So we call it as allergen, okay. So what happens when this allergen enters the body for the first time, okay, the body produces IgE against this allergen, okay. At that point nothing happens, okay. Only IgE are formed. Okay. Now in your body these IgEs are circulating, and then these IgE, they find their receptors. Okay, so because of the specificity, they will find their receptors and they will go and bind to their receptors. Okay, on the mast cells and the basophils, you can see IgE is bound. Okay, now next what happens, so this is when it happens for the first time. Okay, so in that case the person becomes sensitized. Okay, but you won't see any reaction at that time. Now when the same allergen enters into the body for the second time, the allergen, where will it go and bind? It will bind to the FAB region of IgE. So allergen goes and bind to the already bound IgE on the mast cells. Okay, so you can see now when this binding takes place. Okay, uh, it cross links the mechanism. Okay, so it cross links the receptor, and then what happens? A signaling happens. Okay, so a signal is generated. Of course I'm not going to discuss that signaling in detail here. So signaling is generated. Okay, and the signal reaches to the granules. So you can see mast cells are granulated cells, they have a lot of granules here. Okay, now when this happens, a signal goes down and it asks the mast cells to release the granular content. So you can see the granules, they fuses with the membrane. Okay, and they release their content. Okay, now this content, it contains histamines, prostaglandins. Okay, so there are a lot of different mediators. So what happens, these mediators, they go and bind to the receptors on the nearby tissues. Okay, uh, right. So because of this, it causes two very important things. One is vasodilation. Okay, so one is vasodilation, another is smooth muscle contraction. Okay, so what is vasodilation? Vasodilation is basically widening of the blood vessels. Okay, so you can just imagine, so you have a pipe. Okay, and if this pipe dilates, okay, then what happens? So blood is flowing in these pipes, right, in a pressure. Now when this dilation happens, dilations of the blood vessels, the blood pressure will drop. Okay, so the person will start showing symptoms of drop in blood pressure. And then next point is smooth muscle contraction, right? So in your body, the smooth muscles, they will start to get contracted, right? So suppose you have eaten a particular food for which you are allergic, right? So what happens? These histamines and all these things, they will contract the muscles of the stomach. Okay, and because of that, the person will feel uh, like nausea. So there will be vomiting, there will be diarrhea because the muscles are getting contracted. Okay, there will be so the person will accordingly feel the symptoms. So if this process takes place in the nose, okay, then obviously there will be runny nose, sneezing, and all those symptoms, right? Uh, the most problematic thing is when it affects the lungs, right? So then what happens? The smooth muscles of the lungs, they get contracted. Okay. Now, when this happens, the person will not be able to breathe because the muscles are not moving; they are contracted. Okay. So in that case, you will have to give some medicine which does the opposite of these mediators. Okay, so we will talk about that medicine also later on. But at this stage, the main thing is that you understand the structure and the function of IgE. Okay, so IgE, so now you know it has both good and bad effects, right? Okay, so students, any questions? Okay, so with this, we have completed all the classes of antibodies. Okay. Uh, uh, in Cube, you will find one table like this. Okay, so this summarizes all the classes and subclasses of the immunoglobulins. So you have IgG 1, 2, 3, 4, and then you have IgA1, A2, IgM, E, and D, right? So these are all the classes, subclasses of immunoglobulin. You can see their molecular weight here, heavy chain component, right? So we have already discussed this. The normal serum level, okay, half-life of these immunoglobulins, which one is a good activator of the complement pathway, so IgG3, okay, and IgM. So these are the two good activators of the complement system, okay. Which one can cross placenta? So we have already discussed, it's the IgG, right? So you can see uh, IgG only can cross the placenta, present on membrane of mature B cells. So it was IgM and IgD, right? Binds to Fc receptors of phagocytes. So IgG, again, they are good in this, okay. Mucosal transport, so obviously it is IgA, right? And to very less concentration, even IgM, okay. And mast cell degranulation, it is induced by IgE, right? So this is just the summary of all the types and subtypes, and we have talked about immunoglobulin isotype, okay. But there are two more terms, which is allotype and idiotype, okay. And I will not discuss this, okay. So I want some student to just find out the difference between these three, and maybe discuss in the next class, okay. So this is very important to understand. So because see what happens when we will talk about antibodies, so it will just come as a term. So immunoglobulin isotypes, okay, or the allotype or the idiotype. So what is the difference between the three? Okay, so I want some student in the next class to describe these three types. We have already talked of isotype. I am sure you remember when we talked about the five classes. Okay, so I said that these are the different isotypes. Okay, so I want in the next class that you discuss isotype, allotype and idiotype in 2-3 minutes, and then we will move on to the next topic. Okay? Okay, students. So any questions related to antibodies? Okay, no questions. Okay, students, then we can have attendance. So be ready with your cameras. Adish,
Present, sir. Present, sir.
okay, Ayushi,
Present, sir.
Sam,
Present, sir. Present, sir.
yes, Sam, you are not visible.
Visible now.
Okay, okay. So Atoiba is not there. Atoiba, Devanshi, Ishita,
Present, sir.
Ishita, you are not visible.
Am I visible now?
Yes, Janvi,
Present, sir.
Kshitij,
Present, sir.
Manaswani,
Present, sir.
Namrita, Namrita, Nandini, okay, Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba,
Present, sir.
Sanjana,
Present, sir.
Shakti,
Present, sir.
Shivani, Tanvi,
Present, sir. Present, sir.
Present, sir.
Tridev,
Present, sir.
Vivek, Vivek, Yukta,
Present, sir.
Vivek, you are
Sir, I
not
am
visible.
present. I turned my camera on, sir. Am I not visible?
Oh, no, you are not visible.
Okay.
Yukta, Yukta, switch on your camera. And Yuvraj,
Present, sir.
Present, sir.
Present, sir.
okay, okay. Okay, so students, I'm not going to start with the new topic today. So we still have some time. So I think it's a good opportunity. You can ask your questions. So students, whatever I'm teaching is everything clear to you? Because see, I have been explaining in a very simple way. Okay. So I'm sure you are understanding the concepts. Don't try to understand only for your bachelor's. Okay, these things are going to be there for you, with you for a long time. Okay, even in masters you will be if there is a subject on immunology, so that will really help you. Okay, so any questions? So when are you joining the campus for offline classes?
Sir, from next week, I guess.
So it means from 6th.
The 7th of September.
Okay. Why not 6th? 6th is Monday.
Yeah, sir, we don't know that.
Okay. Okay, students, I think we can end the class now. Already we had 2 hours of interaction. So in these 2 hours, we have completed three antibodies, IgE, IgD, and in the previous class, we talked about IgA, right? So we have completed three antibody classes, and let's see what I will start in the next class, maybe with monoclonal antibodies, or maybe I will start with the complement system. Okay, so let's see. Okay, so I'm ending the class now, and if you have any question, you can always discuss in the next class. Okay, I'm sure you will go back, read whatever have been taught, and when you read, you'll definitely have some questions, so you can ask me in the next class. Okay, so I'm ending the class
Okay.
now. Yeah.
Thank you, sir.
Okay.